and some of the pollen grains could survive up to months, like for example Rosaceae, Leguminaceae, Solanaceae. Their pollen grain could survive up to months. But whereas the pollen grain of rice and wheat, they lose viability within 30 minutes of their release. And we can also store them and preserve them uh, in liquid nitrogen at 196 degrees Celsius, and it can be later used for crop breeding programs. Mm. Now let's talk about pistil, so gynoecium and monocar. We already know about monocarpillary, multicarpillary. If they are fused, then it is called syncarpus. If they are free, then it is apocarpus. And pistil consists of stigma style ovary, and ovary is the bulge part located at the base, and then the ovary have locules, and locules contain placenta. uh, Placenta, there are many types of placenta and locules. We have talked about that in morphology in flowering plants, but let's that skip for now. uh, so yeah, megasporangium is also called ovules. It is commonly called ovules, and in number of ovules vary in many ovaries, like in wheat, paddy or mango. There is only one ovule in one ovary, and in papaya, watermelon and orchids there are dozens of many of the ovules. So now let's talk about the ovule, and it is also known as megasporangium. And the ovule is attached to placenta by funicle, and hilum is the junction between ovule and funicle. Uh, Hilum is the reason, or the we can call it as it is mentioned in NCERT. It is the junction between the ovule and funicle. Uh, it is mostly it is protected by one or two layers. Sometimes it is two, sometimes it is one protective envelopes that are called integument. And there is in the hilum side there is micropyle. Micropyle is the small opening in the nucellus, in the tip of the nucellus, and that is called micropylar region. And opposite to micropylar end there is chalazal end represent the basal part of ovule. Chalazal end sits at the bottom and micropylar at the top. This is called anatropous ovule condition. Anatropous ovule. And there, let's talk about nucellus. Nucellus is enclosed within the integument. It is a mass of cells, uh, and nucellus contains female gametophyte, and it has reserve of abundant food material, etc. Blah blah blah. And so now we have our next topic, that is megasporogenesis. Megasporogenesis is the formation of megaspore from megaspore mother cell. Uh, it occurs around the micropylar region, the region of where the nucellus is present, micropylar region of nucellus. And mm, now let's talk about its structure, about how megaspore mother cell is. So it is a large, dense cytoplasm containing prominent nucleus present, and this MMC or megaspore mother cell undergoes meiotic division. And meiotic division results in the production of four megaspores. And now let's talk about the female gametophyte. It is uh, let's yes, after meiotic division from MMC, there are four cells formed, and three of the megaspores get degenerated, and the one left forms the female gametophyte. And there is a little uh, thing mentioned in the NCERT, and it is embryo sac formation from single megaspore is called monosporic development. Uh, Now let's talk about how the division actually occurs. Then the nucellus of functional megaspore divides mitotically. It goes through three mitotic division and forms eight nucleated cells. And yes, this mitotic division are strictly free nuclear. It means that nuclear division is not followed by cell wall formation, and it forms eight nucleated seven cell, and this seven cell structure, and it all that occurs by six cytoplasmic divisions. And in anatropous conditions, three cells move to the roof, and three cells move to the chalazal end. Chalazal end cells are called antipodal cells, and uh, that micropyloidal cells are two synergids and one egg cell. And synergids also have filiform apparatus, and it guides the movement of pollen grain inside the egg cell. And the last two cells that are called polar cells, they sit in the center of the ovule. Now let's talk about the pollination. Pollination, uh, it simply means that transfer of pollen grain to the stigma of pistil. Flowering plants show many different types of pollination. Like autogamy, it is the transfer of pollen grain from the anther to the stigma of the same flower of the same plant. It can lead to genetic depression. Uh, so it is mostly avoided by the plants, but still some plants, clistogamous plants, still do it. Conditions for autogamy are like synchrony in pollen release and stigma receptivity, and stigma and anthers should lie close to each other. Uh, some plants that follows Casmo and clistogamy are Viola, Oxalis, and Camellia. Camellia. As I've already told, clistogamous plants show uh, only autogamy, and there is no chance of cross pollination. Therefore, they sometimes goes to uh, therefore it goes to genetic repression. And now let's talk about gitnogamy. Gitnogamy is the transfer of pollen from the anther to the stigma of another flower of the same plant. And then there is xenogamy. Xenogamy is the transfer of pollen grain from anther to the stigma of different plant. So, about gitnogamy, it is functionally cross pollination but genetically autogamy. Now let's talk about agents of pollination, and the first one is abiotic. Abiotic contains wind and water. Uh, in abiotic, it is mostly followed by wind, and for wind pollination, pollen must be light, non-sticky, and the stamen should be well exposed so that air could easily take away the pollens. And the stamen should be large and feathery, large and feathery stigma, just like we see in corn cobs. It has tassels. The tassels are stigma and style which wave in the wind to trap pollen grains for easy pollination. Wind pollination is quite common in grasses, and so yeah, that's it about wind pollination. Now let's talk about water pollination. It is quite rare in flowering plants like angiosperm. It is very rare. About 30 genres show uh, about 30 genres of monocotyledon, mostly monocotyledons, shows that. There are some water plants like water hyacinth and water lily. Their flower emerge above the level of water and are pollinated by insect and wind. But some plants do show water pollination like Vallisneria. In Vallisneria, female flower reach the surface of water, and male releases pollen on the surface of water, and by the water current they meet, and a fertilization occurs. But there are some other plants like sea grasses. In it, female flower remains submerged in the water, and pollen are released inside the water. It can passively meet for fertilization, but it is very rare. So they release a high amount of pollens. That's why it is not so viable, and many plants left doing it, and now they are stick to biotic agents. And yeah, some bulb pollens do not get wet, therefore they are covered with mucilaginous covering. Now they are done, so let's talk about and yes, uh, pollen or anything flowers or anything like that are not very colorful or produce nectar or produce odor in wind or water pollination, because there is because they don't need anything like that to attract anything, because they are doing pollination by abiotic method. Now let's talk about biotic method, and it can occur through bee, butterfly, flies, beetle, wasp, ant, moth, bird, and some pollen nectar like hummingbird, sunbird, and it is also shown by bats and even among higher animals like lemurs and arboreal rodents or even reptiles. Now let's talk about insect pollinated flowers. They are mostly large, colorful, and they also have fragrant because they should smell nice to attract insects, and they are rich in nectar. And the inflorescence, if the flower is small in size, then they are in cluster to make them look conspicuous. Now let's talk about flies and beetle pollination. So they need foul odor to attract these animals, these yeah, flies and beetles. So that's it about the pollination. Now let's talk about outbreeding device. Uh, most of the plants are hermaphrodite, therefore they are bisexual. Therefore we need some of the methods to prevent autogamy or geitonogamy, or it may cause inbreeding depression. And even plants themselves uh, have some method to avoid autogamy, and some of they are like non-synchronized pollen and stigma release. Pollen release and stigma receptivity are not in sync with each other, and they are placed at different positions. And genetic mechanism is self incompatibility. uh, in unisexual flowers like castor and maize, uh, they are um, mm, they are monoecious, therefore it prevents autogamy. But unfortunately, geitonogamy still happens, therefore they possess geitno and xenogamy. In several species, for example, like in papaya, male and female flower are present on different plant. That is, the plant is unisexual, therefore they only show xenogamy, and auto and geitonogamy are prevented in papaya plant or papaya related plants. Now let's talk about pollen pistil interaction. It is a very mysterious topic because it was recently discovered by scientists how exactly they interact and recognize each other. Actually, pistil could recognize if the pollen is from the same plant or from different plant, therefore reject or accept it. This rejection and exception uh, so excuse me, this rejection and the process of accepting the pollen is a mysterious topic, but now fortunately by chemical components and all that stuffs we have found it. But we have to not study about that, so we'll just skip that portion. So now our next topic is artificial hybridization. It is approach to improve crop programs. So now let's talk about the bisexual flowers. In bisexual flowers, we remove or cut the anther, and uh, we first do the emasculation of anther, then bag the stigma, then dust the desired pollen on the stigma, and then again rebag it. And now in unisexual flower, there is no need of emasculation because it is unisexual flower. Just bag this stigma, then pollinate it, dust the desired pollen on it, then rebag it. Now let's talk about the double fertilization. As we have learned about that there is one vegetative and two generative cells, and if it is in two cell structure, then still the generative cell will divide and form two of its own. So there will be two generative and one vegetative. The vegetative will store nutrients and food reserves, and on which the generative would survive and eat and grow until the fertilization. And now in fertilization, gametes are released into the cytoplasm of cynorgid, and one of them moves to the egg cell and fuse with it to form, uh, and this fusion is called syngamy, and this forms a diploid 2n type cell, that is zygote. And zygote will further later develop into embryo, and the other pollen, uh, the other male gamete will move towards the polar nuclei, the two cells present in between. So they are already 2n type, and other n type of male gamete fuses with them to form a triploid 3n type primary endosperm nucleus, and primary endosperm nucleus will later form endosperm. And this endosperm is a food reserve that stores food for zygote, for embryo actually, because zygote will develop into embryo. And so that was about double fertilization. Now they are post fertilization structures and events. The first very topic of this is endosperm. So endosperm development precedes the embryo development because embryo needs nutrition, and the nutrition is provided by endosperm. So endosperm should develop first and contain and uh, what should I say, like prepare all the food materials or anything like that that is necessary for embryo to grow or to survive. So that's why it develops before the embryo. So there is one more subtopic in it, and that is primary endosperm nucleus undergoes successive nuclear division to give free nuclei. Uh, it is also free nuclear division, so therefore there is a condition of syncytium occurred there, and many nuclei are formed, and this condition can be seen in liquid endosperm of coconut, that liquid portion inside the coconut. Yes, that is liquid endosperm, and it contains multiple nuclei, and the outer layer is solid endosperm, and that outermost layer is mesoderm. So in some plants endosperm is completely consumed, and in the plants where the endosperm is completely consumed are called non albuminous or ex-albuminous, and their example is pea, groundnut or beans. And then there are albuminous seed retain some part of endosperm that is not completely utilized during embryo development, and its example are wheat, maize, barley, custard. And there are other examples also given, in which if the nucleus is persistent, it is called perisperm, and its example are black pepper and beet. So now let's talk about embryo. Embryo develops at micropylar end. We have already know that micropylar end of the embryo sac, where zygote is situated. And the embryogeny is similar in both monocots and dicots. And here embryogeny is early stage of embryo development, the study of embryo, well, early stage of embryo development. That's why it can come in questions in NEET examination or in your board examination, asking about what is common in monocot and dicot. So it will be embryogeny. So now let's talk about dicotyledonous embryo. The zygote can give rise to pro-embryo, and pro-embryo will later evolve into globular, then heart-shaped, then mature embryo. Then a normal dicotyledon have two cotyledons, as it name suggests. Name suggests, two cotyledons. It means dicotyledonous. So the cotyledon above the embryonal axis is called epicotyle, and it forms plumule, or we can call it stem tip, and the cotyledon below embryonal axis is called hypocotyle, and it terminates into lower end in the radical or root tip. So that's all about dicotyledon. Now let's talk about monocotyledon. Uh, as its name suggests, it only contains one cotyledon. It is generally seen in grass families. uh, it is seen in uh, sorry, in grass families. uh, it is generally called, the cotyledon is called scutellum, and the scutellum is generally present to one side, or as it is written in NCERT, to the lateral side of embryonal axis. So the lower or bottom side of the embryonal axis contains undifferentiated seed called coleorhiza, and the upper layer, the shoot apex, contains uh, few leaf primordias, yes, that is enclosed in hollow foliar structure, and that hollow foliar structure is called coleoptile. So that's it all about monocot and dicots. Now let's talk about seed. And but we have already talked about seed. It generally consists of few parts like seed coat, then cotyledon, and then embryonal axis. We have talked about seed coat and cotyledon, and all that embryonal axis, and albuminous, non-albuminous, and perisperm, etc. Let's revise it again. Non-albuminous example are pea, groundnut, beans. No residual endosperm is left, and it is completely consumed, perfectly consumed. In albuminous seeds, some part of endosperm is not completely utilized during embryo development, and its examples are wheat, maize, barley, custard. And in perisperm, a nucleus is persistent. Nucleus is also persistent, therefore it is called perisperm, and its example are black pepper and wheat. uh, so um, now let's talk about how things happen in seed. And when the seed becomes mature, it reduces its water content to become dry, and because of that it can go into dormant state. Or why it can go to dormant state, and why does presence of water can hinder it? Because water contains moisture, and moisture will call many other bacteria or anything like that. And water is mainly the base of any enzyme or catalyzing reactions, or any reactions. It is the base or the surface, or what it is called, the stage of all the chemical reactions. Just That's why cytoplasm is also mostly contained of water. So yeah, where were we? Back to the topic. So it makes itself relatively dry. uh, uh, it loses up to 85 to 90% of the water, and remains 10 to 50% of the moisture by mass, therefore decreasing its metabolic activity and can go into dormancy. and we know that ovules convert into seed and ovary convert into fruit. So if ovary converts into fruit, then the wall of ovary will also convert into the wall of fruit, and that wall of fruit is called pericarp. Then fruit can be fleshy or dry. In fleshy, examples are guava, orange, mango, or anything you can. Uh, we all know what fruit is dry or what is wet or fleshy. And the example given in dry are groundnut and mustard and etc. All that. Uh, so now let's talk about how fruits are developed. Uh, like most of the fruits are true fruits and they develop from the ovary. If the fruit, uh, yes, this is very important topic, and I have seen many questions from this topic in neat examinations. So if the fruit is developing from ovary, then it is called true fruit. And if it is not developing from ovary, not just developing from ovary, just like in some in, for example, apple, strawberry, or cashews, thalamus contributes in fruit formation. That's why they are called false fruit. So now let's talk about parthenocarpic fruit. Parthenocarpic fruits, just for example, banana, they develop fruit without fertilization. Yes, it occurs without fertilization. And how can it form fruit without fertilization? So there are some yeah, growth hormones like auxin and ghrelin, and they induce the feeling. We can understand it as the feeling of fertilization. That's why it goes under parthenocarpic condition. And pollination and fertilization are independent of water. And so now let's talk about the abilities or good things about seed. So they are strategically developed in such a way that they could survive or adapt in many diverse or many new conditions or harsh conditions. They have sufficient food reserve. They have high, uh, uh, what should I say, hard seed coats to provide protection and all that stuff. They have their food reserve, and just like you can imagine, it's a small human living in his home, and he fully packed himself in his home, and he have all the air, good conditions, food, material, and everything. So that's why they can survive for so long. And seed is the basis of agriculture, and dehydration and dormancy is very very crucial. This is the reason why we use seeds so much in agriculture and all that stuffs, because by dehydration and the effect of dormancy, we can store them or use them as food reserve for years without getting them, without harming their quality. So now we have already talked about it, but let's just quickly revise it. There are several very old viable seeds, just like the oldest one. It is called lupin, or its scientific name is Lupinus arcticus. It was found in Arctic tundra, freezed inside Arctic tundra's ice, and it was still viable. And we carbon date by carbon dating, we found that it was above 10,000 years old. 10,000 and still fine. It could still grow. So, yes, this is a very high feat. And the other one we have found is Phoenix dactylifera. Phoenix dactylifera. It is a date plant. Sorry, date palm, DATEPALM, date palm, and it survived for 2000 years and it was still viable. And we don't need to learn about that it was discovered during excavation of King Herod Palace near Dead Sea and all that stuff. it is just useless. Many fruit contain one seed or number of seed. We have already known about that. Orchid are example of many seed containing fruits and etc. uh, plants. And there are some parasitic species of plants like orobanchi, or I don't know how to spell it or how to pronounce it, orobanchi, or and striga, orobaki, orobaki, orobanchi, or striga, orobanchi and striga. Yes. So now let's talk about apomixis and polyembryony. Some mm, some species of Asteraceae and grasses have some special mechanism by which they could produce seed without fertilization. It is a asexual mode of reproduction that mimics sexual reproduction. So this mimicry is called apomixis, and it is also written in NCERT that in some plants, diploid egg cell is formed without meiotic division, reductive division, and develops into embryo without fertilization. Formation of embryo without fertilization, and that is seen in citrus and mango. Now let's talk about in them in detail. Uh, Citrus and mango varieties of some nuclear cells surrounding embryos starts dividing, protrude into embryo sac and develop into embryos. Sorry, we had some misconception here. So yes, uh, in some plants it is example is not given. In some plants diploid cell form without meiotic division and embryo without fertilization, and citrus and mango are not their examples. Citrus and mango have some variety of nuclear cells surrounding embryos and they develop into embryos. In such species of citrus and mango, for example citrus and mango, one ovule contains many embryos. This occurrence of more than one embryo in a seed is called polyembryony. And yes, it is also very important, just like apomixis, polyembryony. Now let's talk about apomixis. Uh, Hybrid varieties of several food material and vegetables are very crucial and good for us. They are very healthy and catches less infection and all that and diseases and things. They are far better than normal plant varieties. So that's why we need them, and for this huge amount of growing population we definitely need hybrid varieties. But the problem here is that if the seed if we collect seed from the hybrid plant which we have sown earlier, then that seed will segregate its uh, normal or original characters and will not maintain the hybrid character. Therefore we have to produce hybrid seeds every year, and it will definitely cost a lot more, and farmers are not so rich to do that. So therefore we have a new method called apomixis. If we make the hybrid plant go or produce aphomitically, or made them into aphomitic plants, then there will be no need of segregation of characters, because there will be no segregation of characters and there will be no need of producing them every year, and it will also be very cheap and suitable for farmers. So at last it has said that in many laboratories we are still working on improving the genetics of apomixis and all that stuffs. So yeah, that was all the chapter, and we have summary also, uh, but I have read that summary and there is not something new or important written in it, but let's just recall some of the things. Anther is bilobe, dithecous, and tetrasporulate, and tetragonal in shape. And yeah, there is one line written here: the central tissue is nucellus, in which the archisporium differentiates. So this is a very important line. Central tissue is the nucellus, in which the archisporium differentiates. Then now a cell of the archisporium, the megaspore mother cell, divides meiotically, and one of the megaspore form the embryo sac. The mature embryo sac is 7 cell, 8 nucleated, and uh, it after everything there are 6 cytoplasmic divisions. So we are 7, well, 8 nucleated, 7 cell, and 6 cytoplasmic divisions. And yes, apomixis is found in some of the angiosperms, especially in grasses. So yeah, that was all the chapter, and it was tiring to do all that in one go. And we have approximately done a recording of 36 minutes, and some other recordings too that I will post in short. So yeah, it was a very long chapter, approximately 45 minutes. We needed 45 minutes to complete it, but here we have done our sexual reproduction in flowering plants. Now we'll do next chapter uh, tomorrow. So till then, good night.